Hi friends, Namaskar. Today we are going to discuss about the sampling procedure in the pharmaceutical industry. And the sampling procedure, why it is to be discussed today. We have a lot of comment in that. Some candidates said that, sir, please make a sampling procedure video. So today we are going to discuss and making this video for the sampling procedure. And this sampling procedure requires or recommended as per the guideline WHO Annex 4 for the pharmaceutical product and requirements. So why this is sampling is required, what is meant by sampling, what is the mechanism of sampling, as per what SOP, which document, we will see all these things in this video. So sampling mostly done as per the WHO Annex 4 for pharmaceutical products and related substance. This sampling procedure requires in the pharmaceutical to verify when particular batch to be released or rejected or used for further production. That time sampling to be required. What is meant by sampling? Sample to be considered as a proportionate or the replica of full batch. So sample means a portion which indicates or represents the complete batch. Many times what happens, we are going to use the raw material in the production, and that time we need to do the sampling, because we cannot trust on each material, how the material came, in which condition it is came, from which vendor it is came. Basically the raw material should be taken by the approved vendors. What is meant by approved vendors? Approved vendors means the supplier or manufacturer who provides the material to the pharmaceutical industry. Mostly the material preferred by the manufacturer, and manufacturer can directly supply to the pharmaceutical industries. During this scenario, when the material is transferred, during that time maybe the storage condition is followed, not followed, what the impact of that climatic condition we have to evaluate. Sometimes this climatic condition also impacts on the product nature. When the consignment received in the company, that time we do a particular portion of the consignment as a representative of that particular batch, and this is called the sample. Who does the sampling? That person is called the sampler. And how to do the sampling, how much consignment, how many containers, or what procedures to be followed, it is all mentioned in the sampling plan. Means how to do, how many containers, who will do, all this mentioned in the sampling plan. Sampling will be done by the sampling procedure. There is a method, standard operating procedure. With the help of standard operating procedure, sampling has to be done. When the consignment received inside the factory, first of all, suppose new vendor is there, we have to do the 3 consecutive batches from that particular manufacturer. All the consignment to be sampled. There are 2 types, like suppose API 100% sampling to be done, because each and every container to be sampled. If suppose excipient 1 plus root n we can do, and basis on that we have to perform the sampling. On this scenario, what we have to do, the sampler should be trained. Particularly the sampler should be trained. He should know the safety practices, he should know the product characteristic, he should know the sampling procedure, he should aware about the safety precautions, all these things. How the samplings to be done? Basically the sampling, when the person go to the sampling, what he will do, he will first select the container or which batch to be sampled. He should check each and every container for the batch number, specification, all the BP grades of the material, product name, material name, all these things, manufacturer name as per his checklist, and verify all the things. Then sample or that particular consignment to be transferred into the sampling booth, and that sampling should be done under the RLF. Under the RLF sampling done, it is the benefit that the material when we sample, that time it will not hazard to that particular person. So it should be done under the RLF. In the sampling booth there is a return and supply provided, so material will not impact it to that particular person. But the sampler should use hand gloves, nose mask, proper gowning, headgear, sometimes if required some nose mask, special nose masks, because sometimes the fumes also can go to the person's inhalation, powder can go into the body of the person, so he has to take proper precautions during the sampling. During sampling, the person should use clean utensils like scoops, sampling rod, sampling container. All these things should be neat and clean properly. If required, it should be done sterile with IPA or some other agents as well. Sample should be collected with the self-sealing bags or glass bottles or PVDC bottles. This type of depend on the material nature, we can use the sampling containers. All these things when we collect and use, that is the representative of that particular batch, and particularly it to be sent to the QC for the analysis. During analysis, the material will be under quarantine, and when the sample goes to the QC, they people will place their level under test level. Once full material is analyzed as per the sampling procedure, sampling testing standard testing procedure, the material founds release, or if the material meets all the standard requirement, the material will be released for the next stage. That time QC people will again push the approved level on the under test level, so the status will be approved material. All this scenario will be continued. In case this is the suppose, sometimes during sampling person has to check the integrity of the pack, integrity of the drum. If the drum is damaged, sealed is broken, it should be considered as a tampered, or that container should be rejected and keep in the rejected area. So all this scenario has to be performed by the sampling person. During sampling, if any abnormal observation is there, he has to report to the quality assurance department. Quality assurance department will check, investigate the consignment, check the consignment, and further decision will be taken. During sampling, the sampler should follow all the procedure and practices. If API 100% sampling to be done, all the containers should be open, there should not be any shortcut. If that practice is followed, your sampling procedure will be done as good as correct way. Suppose if liquid is there, viscous material, properly particular sampling rod to be taken, cup shape, like that all the material will become in the proper way. Also he has to perform stir the material, because if suppose sometimes sedimentation is there, proper sampling will not happen, so some stirring is also required, and he can take the sample and put it into the bottle with closed container. So this is all the sampling mechanism. This is about the raw material sampling. If suppose finished good sampling, the person suppose has to take the material from the batch as a representative sample. If there are suppose 10 drums, 6 drums, he can take from each drum sample. Also, when the batches is going on, that time online sampling can be done, start, middle and end, and that composite sample can be given to the QC as well. Suppose sometimes in the blender also we have to do the sampling during process validation. We will make a process validation video separately, but sampling can be done from the 10 locations, top, middle, bottom. So in that scenario, all the material shall be considered for the sampling purpose, and it will be a representative portion for the batch analysis. Likewise, suppose finished goods, when packing is going on, that time sampling to be taken. That time also suppose blisters, strips is going on, that time also person can check a batch run for how much time it will be run. Suppose the batch will run your 4 hours, that time you can take initial, middle and end, or you can make a calculation based on the batch run, suppose 10 hours, so based on that you can also calculate initial, middle and end, so that particular batch after every strip means foil change, something changes is there or new stoppages is there, all these things will be covered, and initial, middle, end sampling can be done. And that particular sample should be checked properly for with respect to product name, batch number, manufacturing expiry, and it can be considered as a retention sample and can be kept for the future study. Also, there is in the raw material section, there are again sampling methods given in the guideline, like PNR. Suppose P means the vendor is already approved excipient, or the source is already approved, that time we can do 1 plus root n. If suppose the R method is there, R means random, we can do 1.5 plus root n. Then P, in the P means pooled sample. Sometimes for investigation purpose or some special cases we need a pooled sample, that time 0.4 root n we can do sampling methods. So all this way we have to follow PNR method, as well as for API 100% sampling method to be followed. This is all about the raw material sampling, finished good sampling, packaging material sampling, all this about the sampling. Also, if suppose during investigational purpose we have to take a stratified sampling, around 20 locations we can do sampling, and this way we can take the sample for the further analysis. Mostly sample is required with respect to the testing criteria, how much test is there, what test is there. So based on that calculation, sample can be calculated, but sample should be sufficient, that should not be shortfall for all the tests. One more thing, during sampling, if any material is remain, that should not again put it into the batch, that can be discarded. So all this about the sampling mechanism. I hope you understand and this video finds you well. If you like it, share it, subscribe it. Thank you. Namaskar. If you want any more further video, you can comment it. Thanks. Namaskar.